The proboscis reflex behavior of the honeybee. The honeybee, Apis mellifera, is collecting nectar on the flowers of the mustard plant. She licks nectar with her proboscis and carries it in her honey crop back to the colony. When her antennae find and touch a drop of nectar, the proboscis is extended in a reflex action. This proboscis reflex behavior is used in the laboratory for a classical behavioral test to demonstrate conditioning to odors. Honeybees learn to discriminate between odors. Workers of the honeybee are mounted with tape into a metal tube. The tape fixes the bee between head and thorax. Legs, wings and abdomen remain free. A fine glass rod is coated with a sugar solution, then the antennae are gently touched with the rod. The contact of the antenna with sugar leads to the proboscis reflex behavior. Using the same rod, the bee is then rewarded with sugar water. The bee extends her glosser to lick the sugar water off the rod. In the following test, the bee will learn to discriminate rose scent from another scent. A piece of filter paper is saturated with rose scent extract and put into a syringe. The scented air in the syringe is blown through a glass tube onto the antenna of the fixed bee. A red spot, which the bee doesn't see, is lit to show when the rose scent is presented. The bee does not react to the unknown rose scent. Now the scent is blown again onto the antenna, immediately followed by touching the antenna with sugar solution. This triggers the proboscis reflex behavior. As a reward, the bee is allowed to lick sugar water. This unit is called a learning trial. The bee is conditioned to rose scent in this way. Now, when rose scent is blown on the antenna, the bee reacts with the conditioned response to the scent alone. One learning trial is sufficient to trigger the conditioned response in the second trial. In a discrimination test, the odor of peppermint is blown onto the antenna. A green spot is lit as soon as the peppermint odor is presented. Bees which are conditioned to rose scent do not react to the unknown peppermint scent. This sequence of conditioning is repeated once more with another bee. Rose scent is blown onto the antenna. The bee does not react. Rose scent is blown and sugar solution is applied immediately. The bee is then rewarded with sugar solution. Rose scent is blown again. Now this bee too reacts with the proboscis reflex behavior to the conditioned stimulus. Peppermint scent is blown. There is no reaction. If bees are conditioned 3 times with the same scent in learning trials that follow each other, they will react to the scent their whole life. The information of the scent is stored in their long-term memory. Between the large faceted eyes, the bee's antennae are inserted on the head. They are connected by movable ball joints. The proboscis is laid back between the mandibles when at rest. To lick, it is moved forward. This drawing shows a view into the head of the bee. Antenna and proboscis are marked in red. Behind the antenna are the antennal lobes, the first input regions into the brain. Above the antennal lobes are the mushroom bodies, which are named because of their shape. The pair of antennal lobes and the pair of mushroom bodies are also marked in red. The antennae are covered with receptors. Under the electron microscope, one can see many hair-like sensilla and pore plates. These pore plates are for odor reception. The odor molecules penetrate through the pores and excite the receptor cells that lie underneath. Through the pore plates of the antenna, the action potential moves to the glomerular structure of the antennal lobes, which are the initial switching center with many interneurons. Through ascending nerves, the action potential moves to the calyx region of the mushroom bodies. During the conditioning process, the sugar reward stimulus moves from the receptors on the proboscis to the subesophageal ganglion. From here it moves through a neuron called the VUM neuron to the calyx region of the mushroom bodies. Here it is linked to the scent information. The mushroom body is the storage site of the honeybee's memory. The stimulus moves through the output, the alpha lobes of the mushroom body, to the lateral protocerebrum, through the PE1 neuron, an identified neuron. From the lateral protocerebrum, the information moves on through the descending neurons to the subesophageal ganglion. By activation of premotor neurons, the muscle of the proboscis is stimulated, which leads to the proboscis reflex behavior. This is the PE1 neuron injected with the dye lucifer yellow. It is the first identified neuron in the honeybee brain. There is only one PE1 neuron in each half of the brain. This neuron changes its electrophysiological response after only one conditioning trial. We can now understand what happens in nature. The bee touches nectar with her antennae, leading to the proboscis reflex behavior. At the same time, she perceives the scent of the flower. Thus, she conditions herself to the scent of the particular flower species. After 3 such conditionings, she will remember the scent of this flower as a signal for food for her whole lifespan.